Welcome, everyone. Thank you for joining us at Navigating Spatial Biology. Take that next step with Leica Biosystems. Our first presentation for today is Cytometric Analysis of Immune Cell Populations in Archived Clinical FFPE Biopsies for Immuno Oncology Research, presented by Tom Villani. My name is Marie, and I will be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. To ask a question, click on the Q&A box located on the bottom of your screen, type your questions, and hit Submit. The speaker will answer your questions following the presentation. If you have trouble seeing or hearing the presentation, please click on the Q&A box to let us know that you're experiencing a problem. Please join me in welcoming Tom. For a complete biography on our speaker, visit the Biography tab on the left. Tom, over to you.
Hi, everybody, and thanks so much, Maria, for the invite to give this presentation today. My name is Tom Villani. I'm going to be giving an overview of the cytometric analysis of immune cell populations in archived clinical FFPE biopsies for immuno-oncology research. So before we dive in, I wanted to give a little introduction about the company that I work for, Visikol. We're an industry leader in advanced models and tissue imaging. We're focused on improving and accelerating drug discovery and development through our contract research services. We're an innovative and profitable company based in the U.S. And offer research services for drug discovery and development, as well as clinical research. We have a worldwide customer base of startups, small, medium, and large pharma, and a number of prestigious research institutions. Our team is composed of a number of experts in drug discovery, assay design, oncology, immunology, inflammation, liver disease, and many other therapeutic areas. And we leverage that expertise to do several hundred drug screening, preclinical, and clinical campaigns per year. We have dozens of unique technologies and patents related to histology, tissue imaging, 3D cell culture, and other kinds of advanced techniques for biological research. And our two main pillars of our business are our advanced tissue imaging services and our advanced cell culture and tissue model assay services. So a brief overview of the presentation today. So this presentation is going to be focused on how we extract cytometric data from FFPE tissues. And towards that end, I wanted to start with a brief overview of immuno-oncology, which is an emerging and one of the leading paradigms in cancer research these days. We'll touch base with a brief overview of flow cytometry and its applications to cancer research. We'll talk about some of the limitations of flow cytometry when applied to clinical specimens, especially archived specimens. We'll dive into some applications of our FFPE technique for doing cytometry on tissue sections, and conclude with an overview of some of the limitations and the future directions we're going with these techniques. So to start off, immuno-oncology has been a rapidly emerging paradigm in cancer research over the last decade or so. And this is, of course, essentially unlocking the power of the immune system to help fight cancer. Over the past 10 years, there's been an explosion of interest in understanding the interaction between the immune system and how it responds when a tumor is growing. And there's been a huge amount of development towards pharmaceutical strategies to leverage the immune system to kind of help out in fighting cancer. A lot of this started in the 70s and early 80s as we were learning more about the immune system to try to combat AIDS. It was understood that T cells were able to attack cancer cells in some clinical experiments. By the late 80s, it was shown that we could genetically engineer T cells to recognize and attack tumor cells directly. Into the 90s, there was a lot of research done looking at adoptive cell therapy and understanding immune checkpoint inhibition. And by the early 2000s, there was already the first clinical trials to test immune checkpoint inhibiting drugs. The first one was tested in 2000. It was approved in 2011. It's ipilimumab, also known as Yervoy. And by 2012, the first child with leukemia had been treated with CAR T cell therapy. Over the last 5-10 years, there's been a huge amount of developments. Keytruda was approved in 2016. The FDA granted accelerated approval to adalimumab, a PD-L1 checkpoint inhibitor for Merkel cell carcinoma. And with all of this success in a very short period of time, there's been an explosion of interest in research in this space. And there's a huge number of companies right now seeking to modulate the immune response to try to treat cancer. So let's step back for a second and talk a little bit about flow cytometry. Flow cytometry is a critical tool for understanding immunology and really for diving into the population of cells. Flow cytometry is used to quantify cells, but they need to be suspended so that you can run them through the flow cytometer. So in a flow cytometer, we measure the intensity of the fluorescent signal of the individual cells. We typically get them to be fluorescent via immunolabeling. We also can measure the diameter and the refraction/reflection, and this allows us to analyze the populations of cells by selected parameters, such as the intensity ranges, size, and shape. And this gating allows us to do multi-parametric analysis on complex cell populations to really quickly count cells, understand changes in phenotypes, protein expression, to understand the composition of the immune cells that are located in a tissue, understand differences in the cell cycle, understand cell death, apoptosis, activation of immune cells. There's obviously a wide variety of applications that flow cytometry is critical for. And it's routinely used in research, obviously, and also in clinical practice, such as for hematology. So flow cytometry has been a critical tool for a long time. It allows us to take extremely complex populations of cells and essentially come up with a systematic way to evaluate differences in those populations and how they might change or shift due to treatments or the progression of a disease. In Immuno-Oncology, it's incredibly important to understand the immune cell population within the tumor. And so this is typically done in research via homogenization of a culture tumor or a fresh biopsy sample. They essentially disincorporate the cells, decellularize the tissue, and then label it, put it into the flow cytometer, and then can do this multi-parametric analysis to understand the different immune cell populations in the specimens that they're looking at. Determining the relative receptor expression in the immune cell subpopulations is critical to understand the mechanisms of oncogenesis, resistance, and drug effect. And so at the end of the day, the more data that you can get about these cell populations, the more information that you can then uncover about the different factors that contribute to the progression of the tumor itself. So a brief overview of flow cytometry in cancer research. So obviously there's a lot of pros. It's very high throughput and highly automatable. You can do high plex labeling, so it's easy to do 7, 10, 15 labels on a single population of cells. It's extremely quantitative, and you get very high statistical power due to the sheer number of cells that you're measuring. It's got widespread adoption and familiarity. Most immunologists are familiar with how to read these scatter plots. And of course, a major advantage is that you can recover viable sorted cells for further experimentation. Now, there are some limitations to flow cytometry. Flow cytometry can only measure the total of signal. It can't really distinguish between the specificity of staining of internal structures, and sometimes this is important for different phenotypes. Another major con is that you lose all of the spatial information in the tissue. Since you have to disincorporate the tissue, you end up with just a mixture of cells, and you don't know which ones were near which others. And that can be difficulty when you're trying to quantify the changes in the immune cell population in terms of how close the immune cells are to interacting with one another or to the tumor cells. Furthermore, only suspended cells can be put through a flow cytometer. And as I mentioned, you have to disaggregate these tissues using enzymes. And so because of this, you can't use flow cytometry on archived formalin-fixed, paraffin-embedded tissue specimens. It's just not an option. Now, of course, a huge amount of the tissue specimens stored in the world from former clinical trials and research are stored as FFPE tissue blocks. And so enabling a technique that would allow us to do this type of immune cell population analysis on FFPE tissues would be very valuable to cancer research and to research in general. So to that end, we sought to develop a technique to do exactly that. We wanted to characterize the immune cell populations in these archival tissues to take advantage of the millions of archived tissue specimens that pharma clients, research depositories, universities, research hospitals, et cetera, have at their disposal. The most critical aspect is that these stored clinical archive samples have a huge amount of information about the progression of the patient's disease and et cetera. So this represents an extremely large repository of information about disease that we can get access to. So how are we going to do this? So the way that we accomplished this was to adapt the multiplex immunofluorescent techniques that we had developed for doing high-plex immunohistochemistry using digital slide scanning. And we developed some open source data mining and visualization tools that allowed us to develop what we call tissue cytometry. This is essentially doing the same exact kind of analysis where you're measuring the intensity and count of the different cells in your sample, but we're doing it in situ, so directly from the FFPE slide. So how does this work? The approach is essentially sequential multi-round immunolabeling. So the way that this works is by taking a tissue section, labeling with 3 to 5 antibodies, then we do digital fluorescent slide scanning. After that, we strip the antibodies off the tissue using a proprietary reagent that we developed here. We repeat this process over and over until we've covered all the targets on that same tissue. Then we use a computational co-registration algorithm to assemble this into one big mega stack of an image that contains all the different channels. And that allows us to conduct quantitative analysis where we do cell counting, co-localization, intensity gating, etc. So this is a brief overview of how our approach works and some of the preliminary information to demonstrate why we can use it this way. So the approach that we developed involves using a chemical technique to essentially denature the antibodies from the tissue. And this is a very gentle technique that we've developed. It's very rapid, and this allows us to do multi-round immunolabeling. It's essentially like an antibody eraser. And so we can take a tissue. You can see on the far left panel, we have a tissue before staining. Obviously, we see nothing. Then we stain it with our selected antibodies. We can strip the tissue, which gives us back the tissue as it was with no signal. And now to demonstrate that we've fully removed all of the primary and the secondary antibodies from that tissue, we add just the secondary antibody here to demonstrate that indeed all of the primary has been removed and we get no background signal. Then we can repeat with new markers. In this case, we just demonstrated that the antigenicity is preserved. We can stain with the same markers and we get an almost indistinguishable image. One of the coolest advantages of this technique in the eyes of many pathologists is that after you're done collecting all of this fluorescent data, you can still go back and get an H&E slide or cross-validate with conventional immunohistochemistry techniques. And so it makes it very easy to cross-validate this new technique with existing with the existing pipelines. So this is all the critical instrument that allows us to do this is in our lab is the Aperio Versa. And so this allows us to do multiplex multiple channel slide scanning at 40x on these samples extremely rapidly. Usually takes between 15 minutes to an hour for these samples, depending on the size. We get extremely high image quality. They're extremely well suited for quantitative analysis. And this allows us to move directly into our next part of the pipeline. So the most important part of this process, at least from a data science standpoint, is assembling these images together into one continuous image. If you're going to do co-localization of these different biomarkers in the cell population analysis that's required for tissue cytometry, then we have to make sure that the different rounds of imaging have perfectly aligning cells. The way that we accomplish this is by using a technique that's known as computational elastic co-registration. This is a technique that was originally developed to help essentially stitch satellite images together way back several decades ago, but has been readapted for use in bioimaging. And so we actually used for this particular project an ImageJ plugin that's publicly available and free, which essentially takes the DAPI channels of the different rounds of imaging, uses those as a guide, and manages to align these images by slightly deforming the space between the cells to account for the slight deformations that happen when you remove the cover slip and repeat the process of staining. This allows us to get one contiguous image set out of our multiplex image run, and that way we can get a single image with 10, 12, 15 channels, which represent each of the markers that we're trying to look at in this panel. So how does this all fit together? So in the next couple of slides, we'll show you an example of how we've applied this technique to take a look at the patient, at a couple of patients and understand the differences between the immune cell population within these patients. So what we sought to do was develop a panel of 12 targets You can see the targets listed on this slide here. They're mostly for T cells, cytotoxic T cells. We have some memory T cell markers in here. We're looking at proliferating markers. We have pan-CK in there to distinguish between the tumor regions and the non-tumor stroma regions, as well as some checkpoint markers such as PD1 and PDL1. And so we put together this panel and labeled 3 different patients who are all suffering from the same type of breast cancer. And you can see the representative images here displayed with a zoomed in cutout down below. And as you can see from a quick glance, there's not a huge amount of difference between these patients here. And so moving along here, for each of these patients, we get a 13-plex, really, image stack, where we have 12 antibody targets, a channel for each one, as well as the nuclear stain. Here's just a representation of that. The composite image is shown on the left. And in the middle, you're seeing a sequential, essentially, video of the different channels going one after the other on the same image. And off to the right, you can see just a montage of that sequence. So this enables us to get really beautiful multi-channel composite images that look really cool, and they go up on the wall. But now what, right? This doesn't really tell us a lot. Here we're looking at 12 different channels. At the end of the day, there's only so many colors your eyes can see. So these images are pretty, but they don't really tell you that much on their own. The next step of this process involves computational analysis. And so we utilize image analysis software to do cell segmentation and marker colocalization. So we utilize one of several pieces of software. We have several pieces of software we've developed internally. We also use ImageJ as well as the commercially available software Halo to process these image sets depending on the needs. From that image analysis, we get huge data. So we get usually more than 1 million rows per sample, since there's so many cells that we're detecting in these patient tumors. We take that huge amount of data and we feed it through some open source data analysis pipelines that allow us to generate the conventional scatter plots that are typically produced when doing flow cytometry analysis. Just to point out, the kind of data that we get for this analysis is also compatible with FlowJo and other commercially available flow cytometry software for those who are more comfortable with it. We just wanted to emphasize that you don't need commercial software to do this kind of analysis. It's very straightforward and can be reproduced with a basic understanding of Python coding and some of these open source libraries. So what is this overall process of doing the data analysis for tissue cytometry? So first up, with the raw data, we filter by size and shape and co-localize the signals. Then we do some processing such as background subtraction, normalization, log scaling. And finally, we will apply our actual analysis. This is where we're doing the gating and counting, the statistical analyses. And one of the advantages that we have of doing tissue cytometry is we can do spatial analysis. So after we're done looking at cell populations, direct co-localizations, we can dive into which cell populations were located near one another in the tissue. And that's critically important for understanding the roles of the multiple immune cell phenotypes play in the progression of cancer. So here's just a representative example of some selected flow cytometry, or tissue cytometry scatter plots here. And just for a point of emphasizing how much data we're really dealing with, when we've got 12 markers, we have 132 one-to-one primary marker comparisons. And if we want to compare the coincidence of every marker to every other marker, well, that's 12 factorial possible phenotypes. That's about 500 million possible combinations to look at if you want to be complete. And so obviously that's a lot of data. So the best way to do this is obviously systematically and driven by hypotheses. And so we were particularly interested in understanding the differences in the T cell exhaustion between these patients. And so you can see here on the top, we have the scatter plots representing the PD1 intensity is the y-axis, and on the top, the CD38 intensity is the x-axis. And you can see here, patient 2 displays a particular shift in its phenotype compared to patient 1, and we see that same trend with patient 3. Patient 1 has a significantly higher relative percentage of CD3 and PD1 positive cells. This would be exhausted T cells compared to patient 2 and patient 3. Patient 2 and patient 3, we see a much smaller percentage of the total T cell population expressing the PD-1 marker. This goes hand in hand with what we see when we look at the PD-L1 expression in the tumor. And so the figure on the bottom, we see PD-L1 intensity in the y-axis and pan-CK intensity on the x-axis. And here we see we have two distinct populations of cells in our samples. We have a number of PD-L1 pan-CK positive cells, and we also have a number of cells that express PD-L1 that are not pan-CK expressing. And so you can see when we look at the bar graph out on the right, patient 1 had a significant decrease in the, significantly fewer PD-L1 pan-CK negative cells compared to patient 2 or patient 3. Moving along here, we were interested in understanding the tumor-associated macrophages. And so this is where we could start to look at the advantages of a spatial analysis. And so in our study, we can correlate the CD68 positive cells with the nearby pan-CK positive cells and do a scatter plot here to look at the relative expression of CD68 in the region where we see pan-CK positive cells. You can see at the figure on the top, we have a kind of bimodal distribution between our three patients. We have in patient 1 a significantly higher concentration of our macrophages contained within the tumor in comparison to the stroma, where patient 2 and patient 3 show very similar relative percentage of macrophages in the tumor region compared to the stroma. And so to summarize, in patient 1, we're seeing a significant decrease in the tumor-associated macrophages in the stroma compared to patients 2 and 3. Now, finally, the spatial analysis is what really brings this tissue cytometry to the forefront of interest to immuno-oncology. Because of the way that we do this analysis from the actual immunohistofluorescence images, we can take the data analysis and start to bucket these cells into the distance from cell types of interest. And so we were particularly interested here in understanding the engagement between T cells and tumor-associated macrophages in patient 1, since we saw such distinct difference in some of the other, in the T cell exhaustion and some of the other factors that we measured here in patient 1. And so when we generated a histogram of the distribution of the distances between CD3 cells and the nearest CD68 positive cell, we see these figures that are shown in the middle here. And so looking at the top one, we're looking at the distance between your CD3 positive cells and CD68 cells. You can see that patient 1 has a considerably higher percentage of its T cell population in close proximity to CD68 positive cells. And on the graph at the bottom, we're looking at the histogram of distances between CD3 and PD-1 positive cells and the nearest CD68 positive cell. And you can see that trend continues here. We have a much higher percentage of the total population of exhausted T cells in close proximity to a CD68 cell within the tumor in patient 1 compared to patients 2 and patient 3. So to summarize, tissue cytometry is possible by combining multiplex immunohistochemistry, digital slide scanning, and open source data processing packages. The sequential multi-round immunohistochemistry technique that we use allows for 10 to 20 targets to be labeled in a single sample. And this enables rapid multi-parametric data analysis so that we can explore the cell populations within archived FFPE tissue specimens. The differential analysis of cell populations in archived patient tumor biopsies is rapid, it's quite inexpensive, and you get a huge matrix of quantitative data for analysis. We end up with an extremely high statistical power from this type of analysis, since we end up measuring millions of cells in every single sample, and it's a relatively high throughput. With our technique, we can do 50 to 100 slides per day per scanner that we have. We have 2 Versos running pretty much around the clock here, but it's a fairly high throughput technique for histopathology. Most importantly, in my opinion, is the ability to then do the spatial analysis and understand the network of immune cells and how they're interacting and contributing to changes in the progression of the disease. So there are a few limitations to this technique. So compared to flow cytometry, we have a much lower dynamic range. The imagers typically take 8-bit images, and so we're dealing with between 0 and 255 as our possible intensity values. That can make it a little bit trickier to get separation between your cell populations unless you're careful in how you do your data processing. Because a lot of tissues have autofluorescence, this requires either background correction or some kind of chemical processing ahead of time to reduce the background. Of course, since these tissues are fixed, you cannot isolate the cells easily after this analysis. And because these tissues have often been archived for decades, they all require very careful antigen retrieval. And it's well known that some antigens are destroyed by FFPE processing, so not every target will remain available in archived specimens. So next steps, at least on the Visikol side, we're currently expanding this for the analysis of tissue microarrays so that we can offer more complex services for patient stratification. We're next incorporating a number of panels to measure aspects about the extracellular matrix and tumor microenvironment. We're currently in the process of validating a wide library of standard panels for immune markers and other disease markers. And we're continuing to improve our image analysis techniques by incorporating AI where it fits best. So with that, I'll draw our presentation here to a close, and I'll be happy to take any questions from the audience. And thank you so much for your attention. Just about to it.
Thank you, Tom, for that informative presentation. Just a quick reminder to our audience on how to submit questions. Simply type them into the Q&A box and click on the send button. We will answer as many questions as we have time for. Our first question is this technique applicable to live cells or fixed cells?
That's a great question. So it's not really applicable to live cells. We're currently validating the stripping technique and seeing if we can come up with a gentler technique that wouldn't affect the cell viability. So this is mostly for fixed cells and tissues. It does work well with 3D cell culture models as well as adherent cells. And so that technique could be applied in that manner.
Great, thank you. Next question. Can you filter cells by morphology?
Oh yeah, that's a great question. So, since we're doing the image analysis process, we can exclude cells based on size and shape in a similar manner as you can do with flow cytometry. This allows us to pick out cells that eliminate cells that are maybe picked up as mitotic daughter cells or something like that. So we can add that layer of analysis to the pipeline as well.
Great. Next question. Can confocal images be used?
Yeah, actually, so we frequently use this technique both with our Versa slide scanner as well as our confocal microscope that we have on hand. It works quite well with confocal in the same manner as it does with the slide scanner. You can use a sample to get multiple rounds of labeling and do the same kind of analysis. Even you can get a little bit better results with the confocal since you get higher resolution and a crisper image. And so it usually facilitates the image analysis process. Of course, the trade-off is it's slower than the slide scanner.
All right, thanks. Next question, and it looks like this might be the last one we have time for. Is this data compatible with commercially available flow cytometry plotting software like FlowJo?
Yeah, that's a great question. So the output of this data pipeline is essentially a CSV table, and that can be easily imported into FlowJo and other cytometry software that can import CSV data. So it's in a pretty standardized format that can be used with commercially available tools as well.
Well, great. Thank you, Tom. Do you have any final comments that you'd like to add?
Not really. This is it's an exciting technique that we're currently developing towards use in clinical trials and evaluation of drug effect and patient selection. So it's a promising technique that we've got a lot of traction with. We expect to continue to expand this over the next couple of years and find more applications for it as we get closer to the clinic.
Well, a big thank you to Tom, and thank you to our audience for joining us today and for their interesting questions. We will address any questions we did not have time for today and those submitted during our on-demand period via the contact information you provided at the time of registration. This session webcast will be available on demand after the event. That's all for this session. Thank you for joining us, and we hope you've enjoyed the presentation. Bye for now.